Good morning. My name is Marcia Friedman. I'm an assistant professor of medicine at Oregon Health and Science University in Portland, Oregon, and I'm pleased to present our work today on behalf of my co-authors. We are studying the gene expression signature of temporal artery tissue and whole blood in giant cell arteritis. For temporal artery tissue, we use formalin-fixed paraffin-embedded blocks, and for whole blood, we collect blood in PAX gene tubes at the time of biopsy. We collect samples representative of biopsy-positive GCA, biopsy-negative GCA, polymyalgia rheumatica, non-vasculitic cardiovascular disease, other inflammatory diseases in healthy controls. In temporal artery biopsy samples, we also look at skip lesions, and in those cases, we dissect the skipped tissue away from the positive tissue and sequence those two groups separately. These are our results. For temporal artery biopsy samples, the first thing you'll notice is that these patients do have substantial corticosteroid exposure at the time of sample collection. That's true for whole blood samples too. However, we have 8 biopsy-positive sections, 2 skip lesions, 4 biopsy-negative GCA cases, and various control groups. In the PCA plot here in panel B, you'll notice that the P's, which represent biopsy-positive GCA, the pink P, biopsy-positive cases largely cluster together. And the second thing you'll notice is that skip tissue, even though it's histologically normal under a microscope, clusters very tightly with biopsy-positive cases. Biopsy-negative GCA, however, does not seem to cluster with biopsy-positive GCA or with skip lesions. The genes differentially expressed in biopsy-positive GCA are notable for genes related to dendritic cell activation, the chemotaxis of T cells and monocytes, and also monocyte differentiation into macrophages. Pathways associated with differentially expressed genes in tissue include many of the different inflammatory pathways we know to be important in GCA pathogenesis. Of note, interferon gamma signaling is strongly associated with differentially expressed genes. In the blood, we again have substantial corticosteroid exposure at the time of sample collection. The PCA plot here, G represents biopsy-positive GCA. You'll see that biopsy-positive cases do cluster somewhat together, but there is substantial overlap among control groups. And biopsy-negative cases, the biopsy-negative GCA cases, the ends here do not seem to cluster together. Among differentially expressed transcripts, there are far fewer in blood than there are in tissue, and both in blood and in tissue, biopsy-negative GCA does not show any significantly differentially expressed genes relative to control groups. The genes in biopsy-positive GCA blood are associated primarily with toll-like receptor pathways. In conclusion, our biopsies and blood in biopsy-positive GCA cases do show differentially expressed transcripts which reflect the pathophysiology of GCA. We did not see a signal in biopsy-negative GCA tissue or blood, and the lack of a signal in blood compared to biopsy-positive GCA suggests that some of these diagnoses may not be correct, which is a notorious challenge of looking at GCA. We also interestingly found that skipped tissue, even though it's histologically normal, by RNA sequencing looks very much like biopsy-positive GCA. Our limitations include small sample sizes and substantial corticosteroid use, and we are currently working to expand this study by looking at histologically confirmed biopsy-negative cases. So those are discordant biopsies where one side is positive and one is negative, and also larger sample sizes of skip lesions to take a closer look at that group. Thank you for your attention. I look forward to discussing with you soon.